Good morning, everyone. Thank you for hanging in after so many days. I hope you had a very nice countryside dinner yesterday and maybe saw the northern lights. And I hope to light you up with some new ideas, maybe also in this presentation. So let's get it started. Yep. So I'm Theo van der Lee, and this is the place where I work, Wageningen, the campus. It changed a lot actually over the last 40 years. 40 years ago, approximately, I started as an intern in one of these institutes that are now merged. And well, it's really developed for the better. I also took, I think, some changes for me as well in these 40 years, but not for the better, I think. But anyway, I'll try to manage and keep you informed on the current status of our research. So I'm working mainly on the monitoring of plant health and diseases, and this is done actually by two different ways, I would say, by imaging. So we watch satellites and weather information, which can be quite important, as we see now also in the US. We try to forecast epidemics. We go to fields for farmers, and in the second one, the one here on this side, you can see a field that is infested with nematodes. And you can see that actually particularly well from with drone imaging. We try to work also in greenhouses where we look at the produce. We look at seed health, and here you can see an infected cabbage seed with a disease I will come back to later. And we also do microscopy to detect new viruses and to be alerted. Then we also do molecular work, and that has obviously gained a lot of attention in the last years. So we develop single assays that can be run on site, but we also do next-gen sequencing for diagnostics for diseases, but we do full genome sequencing to be able to identify maybe regions of interest in pathogens and also in plants. We try to track actually these pathogens as they are crossing all borders in the world and to keep the agriculture safe in Europe. And we try to modify, let's say, our procedures. Some of our assays are running by the inspection service in the Netherlands. So that is just a community approach to keep things safe. One thing that is important, I've been involved in many map-based cloning approaches for resistant genes, for instance, and then you see that you really need high quality on one side on the genome information. Is there a gene? What could be the importance of that gene, but also on the phenotyping. So if you miss out on some aspects in the phenotyping, you will not be able to match these two things. So we need both qualities on both sides to be able to collaborate and integrate actually these different aspects. All right. I put this slide in. Rick van der Zedde already showed it on Monday on the technology adaptation life cycle. That's actually That's actually the lower part here. And in the beginning, if you have a new strong technology, you see a push from innovators that immediately start working on it. To develop new aspects. You have early adopters, and there you have the gap, and Rick showed it already on Monday. This is a critical phase where actually the expectations are probably very high, but still the possibilities are still a little bit limited before we need actually further developments. So then we go to this early majority and late majority. That's the life cycle. And I think we are, obviously, plant phenotyping is not in one stage at the moment. They are different. And what is critical to note is that this gap is important to cover. And if you want to have a new technology being successful, I think there are three aspects. First of all, there's the push from the innovation. And I think with all the new tools and new possibilities that we have in satellite information, drones, camera systems, machine learning and AI, there's a big push to help developing this technology. And then again, I think it's also important to have facilitators, because you have this curve, this learning curve, and it's a little bit challenging in many cases. So in order to help adopting this new technology, you need actually collaboration, and that's why we're here, I think. to be able to access also to the equipment that is being developed, and also, I didn't put it here, but also access to data. So that will help actually to facilitate the uptake of new technology. And also in the conference, I saw already some nice demonstrators, really some things that are being used in practice, and that helps actually to fit the needs of agriculture and nature conservation. Still plenty of challenges, but we need some of these demonstrators to really kickstart this process. So I think on the whole, we are doing quite well, but there are still some things, and I will show that also, that are in this early phase where we have struggles. So in the Netherlands, we've generated this integrated national research facility for plant phenotyping called MPAC. Maybe some of you already know this facility, as the previous conference was in this location, and you were all able to visit the location as well. And it consists actually of 6 different modules, all fitting specific needs for plant phenotyping. So we have these modules that go down from really detailed all the way to field level phenotyping. These particular key things that are in Wageningen, and I will discuss a little bit more about them, are the number 4, 5, and 6. And this particular items of facility number 2 is merely looking at the plant microbe association, particularly with root systems. And these system 4, I will talk a little bit about more. These are the plants that stay in the same place, and the sensors move over them. And in 5, we have a module in the greenhouse where also plants can be moved to the sensors. And finally, on the field, we have applications with drone and robotics. In addition to facilitate actually the uptake of this type of technology, it's also good to have a little bit more mobile unit. So we bought this Plant Explorer from Innovation that will allow us actually to go different places and to show and demonstrate the possibilities of this technology. So these 5 modules are actually fixed to the location. Sometimes it's a little bit difficult to have people come over and do the experiments. And this one can move around, and that is flexible. Obviously, the drones and the trade seeker, so the robotics in the field, can also move around. What I would like to highlight that is not only the ability to move this equipment around. As Rick already mentioned in this conference a couple of days ago, sometimes it can be difficult to do the experiments in these facilities because of the high-end technology that's integrated in these greenhouses. And the cleaning, it can be difficult to work with pathogens that rely actually on high humidities. So that is why this equipment is also convenient. It can move to the different, this mobile can move to the different locations. Okay. So I will cover 3 topics in my presentation. So I will try to compare and integrate some of these phenotyping methods. I will tell a little bit more about fluorescent labels and how they can be used and applied in the phenotyping. And I will also, if time allows, actually tell a little bit more about the studying of the mode of action for resistant breeding. So the comparison, sorry for that typo there. The comparison of the different methods is actually quite important. As I said, we have different modules. So one of the things that you would like to do is to compare these different, the input or the output that you get from these different systems in a one-on-one basis. So what we did, we planted plants in the field that were monitored by the robotic system, the trade seeker, we monitored with the drones. But we also took plants out of that field actually and measured them in our the Plant Explorer. And then we really have a leaf level, whole plant level, plot level, and a field level comparison. And we did it with multiple pathogens, multiple crops, multiple years. And I will show you just an example on the cabbage that was infected with Xanthomonas campestris campestris. This is a gram-negative pathogenic bacterium. It's causing black rot in brassicas, and the symptoms are actually usually manifested as V-shaped chlorotic or necrotic lesions. So we try to image this from different sensors and see if we can compare across these different methods. One key element, I think, for this type of analysis is the use of a digital elevation model. So normally we measure up to, we get up to 8 or 9 different images at least for each plant. And that allows us actually to build this 3D computer graphics representation of the plants. So using this type of calculations, we can get these plants, as you can see here, nicely separated. And that's important for us because in plant diseases, these diseases are usually not evenly expressed over the whole plant. Typically, the infections are occurring maybe at the lower part in all the leaves or at the tip. So if you're not able to differentiate between these different parts, it's going to be difficult, I think, to get a good assessment of the disease and the disease development. So here you have some images of the drone on the field with the cabbage. So this is the same spot all the time. So these are the different time points, the same pots. You can see here 2 cabbage plants that are growing and also infected with Xanthomonas. And what you can see in the end here, you can see clear chlorosis and necrosis. But it is actually quite difficult to see that in an early stage. And it was already mentioned that with this drone data and also with this trade seeker and robotics field imaging, it can be difficult to get a very early assessment of the disease. So it was already infected around the 10th of August, but symptoms actually became a little bit apparent here at 7 September, but on 22, yeah, that was quite late. It is difficult to use only these very late time points for our analysis. What is also important to note is that you can see maybe the color changes, as you can see that here, but you can also, I can see some changes in the morphology of the leaves that could be informative for the disease as well. It was quite difficult for us to extract the data so far, and the main reason is that we are, you can see the plants are growing, the leaves are all different, so you're looking at different leaves. The diseased leaves can be covered by healthy leaves, and we also had many different types of cabbage. They all have different heights and colors and things like that, so it's a huge variety of diversity, I would say, that you find normally in nature, but it's complicated to work with it. So we're still trying to test some options, how to analyze this data. Let's see. Yeah. So this is the experiment or the data from the trade seeker. You can see that it's moving around. Actually, this is another field, but it moves around also in the cabbage field. And the images that I show on the left are actually from the cabbage. So you can see the individual plants and we can see the growth. But here you can also see, for instance, this is at the same time point and there's a completely different field imaging for the different cabbage plants. So we measure the RGB, the LiDAR, and the hyperspectral data. And we have some assessment on these, but still it was difficult for us to find the disease clearly in the earlier stages. We also did some measurements with the Plant Explorer, and you can see some of these pictures here. So here on the top, you can have a look. This is the color image on this field, and that gives a high resolution, but it also not give you, there's still some difficulties in the analysis there because, like I said, the different types of cabbages, all different types of colors. But what we found very useful is to work with the chlorophyll fluorescence, in particular looking at the efficiency of photosynthesis. So the efficiency of photosynthesis can be calculated after a dark adapted period. You can light the plants and you can measure the amount of fluorescence in the early start and in a later time point. And by this calculating this ratio, you can actually look at the efficiency of photosynthesis. And that's depicted here for the whole plant in the lower part. And now you can see a much clearer effect of the Xanthomonas infection on this cabbage leaf. So this is a false coloring, and you can see the red parts are actually not efficient in the photosynthesis, whereas the green parts are still doing well. You can see that if you correlate a little bit, you can see there are some parts that you may have seen also in the color image, but it really extends far beyond the visual symptoms. And also in this part, you can see it would be hard to differentiate that on the color image, I think. So we provide all types of indexes. I'll not go into that much detail here, but the main thing is actually that the photosynthesis actually works most clear, I think, for this pathogen. The nice thing about photosynthesis is it's quite uniform. I mean, it's important for all plants. So here you can see another example of the detection of a disease, in this case a viral disease, on sugar beet. And you can see some damage here in the RGB image, but here you can see that it extends actually way beyond that, and it's also much more clear and much more easy to quantify. Another example is this Septoria leaf blotch. That's a pathogen on wheat. It's a hemibiotrophic fungus. And you can see here on the left side, you can see the color image. If you are really an expert, you can see these small spots that may be indicative of Septoria, but here you can see it much more pronounced, and it's easier to quantify. So this system of photosynthesis is really quite general. Not all diseases can be picked up quite as well, but in this case we have another example of an oomycete, also an important problem. Bremia is a disease on lettuce, and here you can see again the color image where you cannot observe any symptoms, and here you see the photosynthesis really showing the pronounced effect that it has and on the plant. And here we have the healthy control. You see, these ones are indistinguishable, I think, and these are clearly separated. So this is, I think, a good opportunity to look at these disease processes, and we've used it in many different applications. Here I show the application a long time ago already, where we showed for biocrop science the impact of a fungicide and the protectiveness of this fungicide in a Fusarium infection. So it's non-invasive, it is non-destructive, so you can measure also over time. Although I must say that sometimes the disease expression can also be affected by light conditions. So if you put really strong light conditions and you image very frequently, it can have an effect on the disease development. So there's something that you should check. So first summary, comparing and integration of phenotyping methods. I think for the disease we really need high resolution, and for early detection and quantification we have to be able to look at different parts of the canopy, and the use of photosynthesis and of fluorescent parameters are quite helpful in measuring the disease. There's a complex relationship between humidity and the phenotyping equipment, and this is some of the things that we saw in this first part. Now I'll tell a little bit more about the fluorescent labels. So I start with this introduction on tulip. It's an important crop, and actually Fusarium is a main problem in tulips. It causes this basal rot, and here you can see the symptoms. So it is actually not so easy to score. So therefore what we decided is to use a GFP-tagged pathogen. So we have Fusarium oxysprum, and there's a clear signal here. You can see these are the wild type and the transformant in the GFP signal, and that can be helpful to monitor the disease. So we did that actually in a QTL type of approach, and we mapped a resistant gene. And the nice thing about it is that we've been able to add in the same study 3 additional QTLs based on this GFP imaging. We noticed that the GFP imaging is also giving earlier response. So finally we looked also at the microscopy, because it's non-destructive, you can take some parts from the microscopy, and then we could see that already after one component is actually already visible after 24 hours. And here you see the spores germinating. And in the susceptible, it's germinating quite much faster than in the resistant. And this giving the Physamen head start in the susceptible interaction. So as opposed to the 3 weeks for the visual observation, that you need 3 weeks to do the inoculation, now you can see actually that we could have done things maybe a little bit faster. It also allows us to look at the host and non-host interactions. So here we have a GFP labeled Physamen graminearum on wheat. So that is the natural host, and you can see it's really colonizing the root systems, moving up. But if you have the Physamen oxysprum that I just showed you, and we now try it on wheat, we see that actually it grows a little bit on the root exudates, but is not able to colonize the plant. The GFP can also be used to look at the colonization of plant growth promoting microbes. In this case, we have GFP labeled bacteria that are deposited over here. The roots grow through these areas, and then the bacteria are picked up and colonizing the root. So I think there's many applications for fluorescence, including gene expression studies on the plant, but also maybe if you have a specific expression profile of a particular type of cells, you can then visualize these cells using these labels. There's more than just a GFP, there's many labels actually. So this could be, I think, an important asset. So there are more applications listed here, including controls for transformation, stability, and things like that. And I think many people are using it, and we should integrate it also in our phenotyping equipment. So this second part, so in the fluorescent labels, I think it can really monitor plant pathogen dynamics. It can be used for additional QTL detection, detection of root colonization, and it has near to microscopic levels of detail, and it's quite extendable. So this is an area, I think, where we can maybe see more progress in the future. Here we have, this is a tomato seed that is covered by Clavibacter, which is a pathogen, and you can see that it's not evenly distributed over the seed even. So you can see it's mostly targeted to this area where it will start to germinate, and it can colonize the rest of the plant. So yeah, there's a lot of detail, and I think we should also aim for more detail in order to be able to tell more the mechanisms. So coming back to the last part, there's still a little bit of time, so I'll just try to explore on that. So we have, we try to work with these recombinant embed lines. We have wild types that are resistant, in this case to botrytis, and we've integrated parts of this wild type material into a commercial variety. And then we would try to see if we can differentiate these the resistant levels during the infection of botrytis. So this is the protocol that we used. We look at the RGB, we look at the efficiency of photosynthesis, chlorophyll content, and we also have hyperspectral analysis performed. So botrytis cinerea is a necrotrophic fungal pathogen. It has a short biotrophic phase. It has a wide host range, and it's gray. It's called also the gray mold, and you can see it. Oh, sorry. You can see it also here. That is producing a little bit of grayish color, but it's not so easy to differentiate. So we'd like to know if we can use this new equipment also to identify QTLs. And we hope also to improve actually the classification of these QTLs. So the first thing that we looked at is lesion size, an obvious thing to do. And we can see that there's clearly a difference between the commercial cultivar and the resistant variety in terms of size. So that is easy and logical to do. But we also found that the shape is actually different. So here we have the spread of the disease in the commercial tomato cultivar. And we see that it's really targeted towards the different specific parts of the plant, the veins, and it's really irregularly shaped. as opposed to the wild variety, where you can see actually that it's really rounded up quite nicely. And so we use this calculation to get an idea on this containment actually of the disease locally. And then there's also difference in lesion types. So it's not only the size, but you can see in different aspects actually that the color is also different. And we think these 3 different parts could indicate maybe different components of the resistance. So this is, we try to integrate these type of things in the other practices, so with the genome sequencing and the mapping, and hopefully we'll be able to identify a new resistance gene for complex traits, such as these quantitative traits for botrytis. So in summary, I think we have the integration of the phenotypic methods. We have used the fluorescent labels, try to study the mode of action, not only for fungicides but also for the resistant genes that we find in wild species particularly. And we would like to integrate it a little bit more with other expertise. I didn't cover that much today, but obviously it's integrated also with the qPCR. We can quantify the fungal biomass in this infected tissue. We can look at RNA-seq to look at the expressions of different components, and we look at the genome sequencing. And with this, I would like to acknowledge the people that were mainly involved. It's a large team because many different aspects were covered. And in particular, I would like to give credits to the PhD student, Laura Groeneberg, here on the right side, and also the postdoc Marie Duhano here that did most of the work but couldn't make it because they are just they are young mothers that like to stay at home a little bit, which is quite understandable, I think. And here are the companies that are supporting this research, particularly looking at the phenotyping of traits for resistance and integrating that into their breeding practices. And with this, I would like to thank you for your attention.
Oh, thank you very much for the nice talk.
Yep.
About how to detect the diseases and bring it back into breeding. So we have time for a few questions. So where are the questions? If there's no question, oh no, back there. Yeah.
Hello, great talk. I have a question about the in situ imaging of the tulip roots and the pathogen, GFP pathogen, and what is it? Is the imaging done with a hydroponic setting, or which exact imaging method have you used?
For the GFP imaging, you mean?
Yes.
Yeah. So there's specific channels actually. So it is specific light and specific filters to visualize.
I saw that you could image like the shoots and then roots and then the infection through like exudates.
Yeah, so that's correct, because we have one camera system and a filter wheel, so we can actually put everything pixel on pixel in shutter. So what I showed is actually the image of the GFP on top of the color image, because everything it's one camera with a filter wheel. So all the information that we have from the GFP from the photosynthesis and for the RGB is all stacked actually in on top of each other, pixel to pixel. So we can just put it on top. It's just a visualization.
It's on the riser box. The imaging was done in the riser box, and then.
So it was actually done in there's agar. So we have these are the big plates, and we basically fill half of it in the diagonal and put the seedling in the middle. And then they are monitored. Yeah.
Over there in the middle.
Hi, thank you for the talk. Very beautiful images. So I have a question on the chlorophyll A fluorescence part that you showed at the beginning. And you showed that you had some OI-JP curves as well. And besides the palm measurement, have you found like good differences with the OJ parameters, especially in the field, since they relate more to photosystem 1 application as well? And probably I didn't catch this, but did you use the portable instrumentation in the field, or you took your plant to outside?
Yeah, no, we take the plants from the fields. So actually they were just in pots, so we could just remove them and bring them to the lab. But then we returned afterwards again. Yeah.
And you measure both the OJT and the palm with the same instrument?
Yeah, normally we use the palm measurements, but sometimes we also use different methods. But basically the results are very similar. Yeah, because we're mainly looking at the differences within a leaf. So we're not that much interested maybe in the absolute values. It's mainly the pattern that we like to see.
So you haven't found anything that particularly striking from the OJT, let's say? Like it was repeatable with the palm?
Yeah, it's repeatable, yeah. It's actually very, very similar.
All right. Thank you so much.
Okay, so the last question here in the middle.
Yeah, Dr. Vanderlei, thanks very much. Fantastic presentation. So, the photosynthesis, a signature is quite interesting, of course, and you can see diseases often even earlier than the human eye. But of course, it's not often non-specific. I think many diseases will cause probably impact on photosynthesis. So has there been thought to perhaps combining with other data sets, perhaps with the weather at the time, or maybe the pattern of the particular the signature, so we can perhaps identify which disease it is, because in the end the farmer needs to know which disease it is. Yep.
Yeah, so at the moment we do the inoculations, so we're pretty sure what is there, and it's mainly, like I said, the photosynthesis is mainly in the lab. So we're not going to advise farmers, but I know that there are some people that are doing these experiments outside, and they measure photosynthesis outside. They are still developing things, I would say, and the main problems in their hands is mostly to extract the information from the right crop, because there may be weeds and things like that, so there's a lot of complications there. But I do think it's also possible to use it for field situation, but for really large-scale monitoring, I think it's not going to be implemented. And I do see that, I show some different aspects, and most of them are really distinct. But you have to, again, classify, let's say, and develop an ontology similar to what we have for the fungal diseases in the end score on the RGB image, let's say. You should do something similar for that, for photosynthesis, perhaps. And indeed, if you combine it to the region, to the host, to the timing and climatic conditions, that's logical that you implement that also and integrate it in an assessment. But this is mainly done in the lab, so we know what we have.
Well, great talk. Just a question about, these assays that you must perform, they're mostly manually. And what would be the challenge to bring this now to high throughput so we can really make sense of the complex biology behind that?
Yeah, indeed, this is quite a hard labor sometimes to do all these inoculations. We've automated some of them. In the greenhouse, for instance, we are able to automatically inoculate the plants, for example, monos, so that helps a little bit in the throughput. But in the field, there's also possibilities, I think, to automatically inoculate, at least with the traits. So there is potential, but it depends a little bit on the topic of study. Because if you have obligate biotrophic fungi that I also showed, it can be hard to get enough inoculum to do this automatically, because you need a little bit higher volume, and then it gets more complicated, and it's back to the handwork. So our key thing is actually to work on the processes, the mode of action, in a small scale experiment, and then implement that in the larger scale when we know the mechanism. And hopefully then it will not, if we know the genes, if we even can transform the genes into the the crop. It will be it's just a validation that you have to do in the field. That is the approach that we try to follow. Does that answer your question? Yeah.
Is there another question? Oh, up.
Thanks, Theo, for your speech. I mean, beside the sensing of the disease and the spread of the disease, I mean, are there a kind of mechanistic models that can help you to describe the dynamics in space and time at that scale of the disease? And how do you see the link between the observations that you get from your high throughput phenotyping experiments in such kind of models?
Yeah, that's a very nice question. And well, obviously there's other factors that we consider. I already mentioned, so is the plant, the age of the leaves, things like that. So if you have crop growth models, for instance, that can help, I think, to get a better grip on this disease expression. So we are working on these crop growth models in some of our crops, and that could be implemented perhaps in a later stage. We have, in this case, there's a lot of challenges, mainly because you see so much genetic variation, and I didn't hear that much in most of the talks, but there can be substantial differences between genotypes, and that's something that you have to take into account as well. Hopefully in the future there will also be more information on the modeling of 3D, for instance, for the different genotypes, and implement different growth models for the different genotypes, and then perhaps that can be it can be integrated, but it takes time. I would say.
And eventually can be introduced in such kind of models the different susceptibility of the genotypes in the propagation at the leaf, but also at the canopy scale of the diseases. Do you think it's possible using high throughput phenotyping in control conditions? Or
yeah. So if you have that information available in your metadata, I think it can be used. It's well known actually from literature that some of these infection processes, for instance in the botrytis, can happen quite early in the stage, like for instance in the flower, but then there's a long latent period until fruits start to develop and things like that. So there are specific processes that occur during the development of plants, and you have to take that into account. If you have different genotypes, perhaps you will have earlier development of fruits, and then also the disease will be impacted by that. So also the hormone developments. Yeah. So there are a lot of things connected. At the moment, I'm mostly focusing on the disease, capturing really good images, high quality images of the disease process, and trying to find the genes that are responsible for the resistance and susceptibility. And the next step, hopefully it will follow maybe with this new machine learning and AI type of technologies. But I think it surely will depend also on high quality data to start off with, and the literature that is also available. Yep, thank you.
So is there any...
Hi, it was a great talk. On the first day here, I heard that X-ray imaging can damage the plant. And now I heard you say that taking pictures can also affect the growth and the diseases on the plants. What can you tell me more about it? Some insights? Is it because of the illumination, or why did you say that in particular?
So if I get your question right, it's about the 3D structure and the impact on the disease? Or
no, it's about the imaging actively the plants can affect both the growth or the disease spreading.
Yeah, that's a good question. Yeah. So the light actually has a lot of effect on the plant, and the measurements that we do, actually we saturate the plant with light in order to capture the photosynthesis. So we wanted to do some cool movies, obviously, showing the effect on of the progression of the disease. And then we found out that actually it was quite remarkable that we had delay of disease expression in these plants. Luckily, my PhD student also had control samples which were not exposed and that were actually in similar conditions all time. And we saw a pronounced effect actually on the disease development. So that's why we started to go a little bit lower resolution, a little bit higher distance, and we are measuring a little bit less frequently in order to still have, let's say, the similar disease progress compared to our non-measured control plants. But it was a pronounced effect, and I think, there are also people interested in this because perhaps with the light, changing the light condition or with flashes, it will be possible to reduce the infection and the disease progress.
Any further question? And maybe from my side, so how far do you think are we to not just detect that there is a disease, also in the field, and know what kind of disease it is?
Yeah, that's a difficult question because it depends really on the crop. I think in most cases, most of the crops are in particular region are usually affected by the same disease. And sometimes it switches a little bit after a few years. You can maybe have something new in. But if you have, for instance, potato, you will likely have potato late blight in the end of the season. So many things are already known. And there may be some curious disease that we don't have enough images for to really call it in this way, but probably they're also not the most important one. I think it's important to separate, let's say, the top 5 or top 10 diseases per crop. And then depending on the season, depending on the symptoms expression as I showed, and depending maybe on the resistance that you have in your crop, and maybe spray use, I think it will be possible to capture those quite well. But there can also always be new things, and then you have to start again. Yeah, but we noticed actually we have inoculated our plots, and we noticed actually some other diseases that came into our plots. Yeah, we could directly tell this is not, it looks really irregular. So we could directly tell this is not the inoculum, this is something else.
Yep, thank you.